So I'd like to talk to you this morning then about a bunch of tools that we've developed over the years to help us with the initial processing and QC of large data sets, particularly sequencing data sets, but the same principle applies elsewhere. And the rationale for this was underlined quite nicely recently by an article that was pointed out to me, which was around tools that have been built to try and help with the detection of COVID. And the slightly depressing conclusion was that a lot of people had built machine learning tools to try and do this type of detection, and really none of them worked. But the interesting sort of side from this was why. In the end, when we're trying to build these kinds of predictive models and tools that work off large data sets, we are looking for signal within our data. And the tools themselves don't really care where that signal comes from. Ideally, we would like that signal to be the result of the biology or the disease that we're studying. But if other confounding factors come in to provide signal, the tools will happily pick up on that. And in the case of COVID, really trivial things were actually driving the models that were built. So, for example, non-COVID patients were mostly children, COVID patients were mostly adults, and you could tell those apart on the scans that were fed in. People who had COVID were often scanned lying down, whereas the people who didn't have COVID, the control group, were often standing up. And again, you can tell that apart on the scans. Even really trivial things like certain hospitals had large numbers of COVID cases and other hospitals had relatively few and provided more control cases. And those hospitals annotated their data and they used different fonts. So the models were actually picking up on the font that the names were written in on the scan and were predicting COVID on that basis. The upshot of this was that the conclusion here was that, yeah, everyone wants to get into the sexy side of building predictive models, doing exciting statistics, but those can be completely undermined unless you solve the slightly trivial, boring problems at the outset to ensure that you have data which is valid and without confounding factors going in. And that's where I want to focus my attention. So the 2 points that I'm trying to get across then this morning are, firstly, in your initial data processing and validation, you want to check everything you think you know about your data, because it's surprising how often something turns up that's unexpected and would confound your analysis later on. And secondly, when you get past the processing and into the analysis, it's important that you take an explorative approach because, again, things will come along which you didn't expect. Sometimes those can reveal interesting biology. Sometimes they can just be problems to deal with. And the combined set of these will give you better outcomes at the end. So for the QC initially, I'm going to talk about a number of different projects that have been developed. Some of these have been around for a few years. Some of them are very new. And all of them are things that we now plug into our standard processing pipeline for large sequencing data sets, both ones we generate internally and those where we're reprocessing public data. And the things that we're trying to validate are things like, are my processing metrics sensible and equivalent across all my samples? Then things like, am I working with the right species? Am I definitely getting the samples I expected? If I'm in the right species, am I in the right strain or individual or cell line? And then does the data that comes out from this sample look like the type of sample it's supposed to be? And we can address all of these in an automated way. Looking at the first of these, I'm going to talk about a program called MultiQC, as written by Phil Yules, who used to work with us a while back, now has his own group in SciLife Lab in Sweden. And MultiQC, if you've not seen it, it's been around a little while now, but it's super useful. So what it does is it goes through a folder where you've done a whole bunch of processing. So it looks at all of the output files from a processing pipeline, and it hoovers up all of the QC metrics or processing metrics that it can find from a wide variety of programs and presents them together in a coherent report. The idea with this is that even if you don't necessarily know what you expect or what you're looking for, just by comparing the metrics you get across a wide number of samples, you can spot outliers and know where to focus your attention. So in this case, we have a graph where it's looking at the total amount of data that made it through a processing pipeline, in this case from a data set that should have been lots of different mouse cell lines. We can see that there's some variability here, so some have more data than others. But down here, you can see that there are two samples that didn't fail. They've made it through. They have a couple of million data points, but they look wildly different to everything else. So it allows us to focus in on what these things are. Once we know where we're going to focus our attention, we can start asking questions about those samples. The first question we want to address sort of systematically is that we're definitely working with data from the right species. And for that, we have a program called FastQ Screen. What this does is to sample a large data set, pull out a small subset, and then search it against lots of different genomes. We can then customize the set of genomes we've got. We have quite a big set because we work with a lot of different species, and we get back a summary which looks something like this. So here we can see that we've tested many different species, and we can see where the results have come from. So in this case, all of the blue parts of the bar here, those are the ones that only matched against one genome. The red or orange parts matched potentially against multiple genomes. And we can see that all of the unique matches come from mouse, which is great. It's a trivial thing, and most QC should be trivial. We should just be able to validate and move on. In the example I showed before, when we had the large collection of cell lines that should have all been mouse, the two that lost most of their data, it turns out weren't mouse at all, they're rat. They look exactly the same. They were purchased from a supplier who said they were mouse, but finding stuff like this allows us to clean up, remove the unnecessary signal from the data, and get better results at the end. Even once we're sure that we're working with the right species, we might still not be working with the right individuals from that species. So for mice, we're generally working with lab strains of mice, and there have been numerous reports in the literature of studies that have been confounded because the strain of mouse that was used wasn't the one that people thought they had. These things pass between labs, they're bred within an individual institution, and it's easy for things to get swapped around. We have the ability to validate this, though. So a while back, the Sanger Center did a big sequencing project where they sequenced all of the commonly used mouse strains and then produced a high-quality list of variants for each strain. What Felix Kruger in my group did was to take this and expand on it to pick out the diagnostics SNPs which would be associated with each of the individual strains, and then has built a small program called Restraining Order, which you can feed a sequence data set into, and it will now validate that against all of the possible strains and predict which strain you have. This, again, is trivial in most cases. It just validates what you think you know, but often it catches things out. So here's a standard result from this. So this is a purebred strain, and you can see that in the table that it comes out, these are all the strains that it tests, and this is the compatibility with that. So you should see that the strain you expect has a very high value. In this case, this is a C57 Black 6 mouse, and it's very compatible with that. It's even more clever than that. So often you are not dealing with purebred strains, you're crossing different mice, and crosses are even harder to validate. But again, from this data, Restraining Order can actually do that. So it will say which hybrids are most compatible. So in this case, a 129 Castaneous hybrid is the strongest prediction, and it can even tell you what the proportion of the two strains is. So this is likely to be a first generation cross because we have about half and half of each strain. You can predict whether you're F1, F2 crosses. And again, this is stuff that can just reassure you about the provenance of your data or spot problems. And anytime you introduce one of these things into your QC, you're going to start finding problems. So this was a report from something that was supposed to be a pure Black 6 mouse, but when we run it through the program, we can see that Black 6 actually is really not compatible with the data in here, and this is the wrong strain. Finding this stuff as early as possible saves you time, saves you effort, and saves you money, and gives you the ability to fix this. Now, mice are easy because they're back-crossed, they're purebred, and we know all the strains. Humans are more of a mess. But the same principle applies, that we know something about many of the cell lines that we use in humans, or it might be individual patients that we know about. And we also know of kind of historical cases where mixes in cell lines or contamination from other cell lines are actually driving effects that we're attributing to biology or disease. And we want to be able to find those. For human data, we have cell line Sleuth, which takes on the same sort of approach as I showed before in mice. We can do this. We're showing this as examples of SNPs taken from the Cosmic cell line database. But if you had individual genotypes that you'd sequence from patients, you could equally plug those in to validate the source of data sets from that. We can plot this out visually in this case. Now, humans are a bit more of a mess because you don't necessarily have fully penetrant variants in here. We can have partially penetrant things, heterozygotes. So we look at this visually. In this case, we tested a bunch of different cell lines. So here are the SNP sets from different cell lines. And from our data, we see which alternate alleles actually turn up. So here we would be fairly confident to say this was the Colo 205 cell line. When we started checking this more widely, we found cases like this, where this should have matched one of the cell lines, and we really don't see anything. So we don't know necessarily what it is immediately, but we know that it's not any of the cell lines that we've tested here. And with a bit more searching, we can find that actually this came from another cell line that we'd not looked for before, this HD29. So something that will help the scientists reassure them or allow them to go back and correct mistakes at an early stage. The final sort of metric I'm going to talk about is the newest one that we've just introduced into our QC pipelines now, and it's the work of Crystal and Katavia. So they've been looking at the sequence composition of sequencing libraries generated by different library preparation techniques. There are a wide variety of different sequencing libraries you can make. So RNA-seq to measure expression, ChIP-seq to measure protein localization, ATAC-seq to look at accessibility, bisulfite sequence to look at methylation, lots of different library types and lots of things you can measure. And what they noted was that the different library types have different biases in the composition of the base calls that come out of the libraries, linked to often the way that these libraries are prepared. So another sanity check that we can now do is we could use the composition of the library to compare to what we expect for that library type to make a prediction of whether we've got the right kind of data for the experiment we think we're doing. To do this, what they did was to go through the public data sets. So they went through the geo data sets of public sequencing data for lots of different libraries and calculated the composition, fed them into a small machine learning model, and then projected that into a graph like this, where you can see that just based on the composition of the libraries, we can quite nicely separate the different types of library prep that we have. Once we have this model, we can then take any new data that we get, calculate the composition from that, and then project it into this model to make a prediction of what type we think it is. So for any new library we produce now, we can make a graph like this, which is just the confidence that the model has for the type of library we think that data came from. And in most cases, it's finding the expected type, but if any problem comes up, we catch it very early. An interesting side effect of this work is that they looked in more detail at some of the samples that have been submitted to the public repositories. And in particular, they found interesting things when they looked under the RNA-seq designation in GEO. So this should all be expression data. But they found that when they put this through their model, that it didn't form one coherent block, but actually you got lots of structure within it. And looking in more detail at that, they find that actually, although everything is described similarly in here, there are actually multiple types of library conflated. So we have some that are tag-mented libraries, some that are strand-specific, some that do pre-amplification, some that are single-cell libraries, or what they termed weird stuff, which are things that are related to transcription, but they're not really RNA-seq, but there really isn't any other label you can place on them. So if we're building models that are going across large swathes of public data, we need to be aware of things like this, that just because it's labeled the same in the database doesn't mean that the data is all exactly derived the same way or that will behave the same way in a subsequent model. So the upshot of all of this is that when we're processing our data now, we're running these kinds of tests automatically. We release this software so that anybody else can do the same thing. And in most cases, it's a trivial step of just looking down and saying, yep, that validated, and that did. But if any problems come up, we can catch them as early as possible. The second part I would like to step onto then comes beyond that initial processing. So once you've done the automated processing of the data, you're getting into the analysis. And many companies are now trying to encourage the automation of the analysis of data. So things like the Dragon system from Illumina, projects like Galaxy have been around for a while. And the idea is that with these, you can build quite complex pipelines running a number of individually very good analysis tools to quantitate, normalize, do statistical analysis and evaluation of outputs in a very large and very black box where you get to see the results at the end of this. And this is something that we've been concerned about for a while, because although the individual components of these pipelines are very good and very well maintained, there's an awful lot of things that can be hidden by this kind of approach. So the final bit of software I want to show you is something that we've been developing for several years now, which is a graphical visual analysis tool called SeqMonk, which can do the same sort of processes as these sort of sequence-based pipelines, but does it in an environment where you are encouraging interaction with the data, presenting visual results in front of you and making it easy to kind of dig back into the data and explore it. And I just want to illustrate the value of this by looking at some of the things that we would get people routinely now to check alongside the biology that they are initially interested in. So some of these are relatively trivial. For example, we think we generally know what sex the samples that we're dealing with are, but it's surprising how often things don't quite pan out. So here's an example of a data set where we were told when we were given this data set, these are all male samples. It's pretty easy to check if something's male. We're doing this in two ways here. So firstly, we're looking for data coming from the Y chromosome, which you should have if you're a male and you shouldn't have if you're a female. Also, because this is gene expression data, we can look at the EXIST gene. So EXIST is the gene that inactivates the second X chromosome if you're a female. So one of them is silenced, one of them is active, and EXIST is the gene that does that. So if you see signal from EXIST, you're likely to be female. And just by looking at these two regions, which is a trivial operation, like 5 seconds in SeqMonk will bring up these two different regions. We can see that in their supposedly all male samples, we definitely have a female sample. It has the EXIST signal and it doesn't really have much on the Y chromosome. And we also have a mixed sample. So yes, there's EXIST signal, so there's definitely some female in there, but we also see that there's definitely Y chromosome signal. So 2 samples in a relatively small study is quite a lot of noise to add into this. Spotting this early, kicking it out, gives you better results and outcomes. This is a relatively trivial thing. Yeah, it's a QC check. We find a problem, we kick it out. Some of them are slightly more insidious in some ways. This was an example of a study where they had 3 different biological conditions. And again, we did the sort of standard validation. We think, are these male or are they female? And it turns out that whilst all of their samples for condition 1 and condition 2 are male, all of their condition 3s are female. So although we can go ahead and analyze this data, when we come to drawing conclusions based on what changes in condition 3, we can't be sure that those conclusions are really linked to the biology that they were trying to explore. They could equally just be that they're comparing a female sample with a male sample. The two are completely conflated. So there's not a lot we can do to fix it at this stage, but it's something that firstly they should have thought about more before they got to the design. But if this does happen in the data, we want to identify that so we can put the appropriate amount of confidence into the results that we have. When we're analyzing public data, we are particularly encouraging our scientists to be critical of the provenance of the data that they get from public repositories. You don't know where it was come from or how it's been handled. All you're doing is reading the paper and assuming that what they claim is correct. And it's surprising how often things don't quite pan out when you go and look at the data behind. So something as trivial as this paper did a knockout of 2 genes. Okay, that's the point of this. They're trying to investigate the functionality of this system, and they knock out these 2 genes. When we go in and look at the data, we can go and see the 2 genes, and we can see that in both the knockouts and the wild types, these genes are both present. Okay? We looked up the way that this knockout should have happened, and it should be a complete removal of the whole gene, and neither of them has gone. So whatever is being reported in here, or whatever looks different between these samples, it's not because of the knockout, and we just need to move on and do something else with our day. That's obviously an example of something that fails, but sometimes things fail in slightly more specific ways. So as another example of a paper that knocks out a gene, here's a paper that knocked out a gene. Here's the gene that knocks out, or is supposed to be knocked out, and it looks more encouraging. So here we have a bunch of samples where we can see signal from that gene, and a bunch of samples where we don't. Great, that's what we're expecting. However, in this case, when we go and look at the annotation that's associated with this, we find that these samples are annotated as wild type, these are annotated as knockout. So although we'll do the analysis, we'll find all the differences, we'll get all the conclusions completely backwards because they've been mislabeled. Interestingly, in the paper, they actually get the labeling correct. The claims in the paper all pan out, it's all fine. It was when the data was submitted to the public repository that the metadata was switched. So a bit of sanity checking and validation allows you to spot this and get the correct result out. As my final couple of examples, most of the things that I've shown you are kind of bad cases, things where something went wrong, okay, where we needed to fix something. And okay, that's the nature of QC, that's what it's going to spot. But particularly in this sort of explorative sense, there are some potentially really positive things. So finding things that you might otherwise have missed, okay, and that aren't necessarily expected when you thought about what you would see in the data set. So I'm going to finish with 2 quick examples of this kind of positive adjustment that you can do with an explorative approach. So this is the first one. So this is an example of an RNA-seq comparison. So these are scatter plots of 2 conditions on here where each dot is 1 gene and you're measuring the expression level of that gene. Okay, so anything that's showing the same expression in the 2 samples should sit on this sort of diagonal that we have on here. We are using statistical tests within SeqMonk based on the DESeq2 package, which is the most widely used package for differential expression and is a very good and very well sort of constructed and maintained package. Okay, we use it all the time. But it makes assumptions about your data, as do any package that does processing or analysis of data. And sometimes those assumptions don't pan out, and if they don't, then the results that you get might not be correct. So one of the assumptions DESeq makes is that it expects that in any comparison of 2 samples, you're going to see about the same number of genes increasing in expression as decrease in expression. Okay, that those changes should be roughly symmetrical. It doesn't have to be exactly, but roughly. And most of the time that's true, but not in this case. So in here, when you look at the comparison, you can see that most of the points which are falling away from the diagonal are falling underneath. There is a strong directionality to the change in this particular piece of biology. And the side effect of that is that the normalization that was done on the data didn't really work very well. So the normalization should place the genes which don't change in expression exactly on this diagonal. And up here, if we look, you can kind of see that there's a straight line here that falls a little bit above. And the consequence of that is that when we start predicting differentially expressed genes, we get what we see on the right, that it says, okay, yeah, there's a bunch of things that go down, that's fine. But if we focus on the red ones that go up, we can see that a lot of genes are predicted to go up in expression, including all of the really highly expressed genes, which doesn't seem likely. It's more likely in this case that we have a mistake in the normalization. If we can spot this, we can apply a relatively small adjustment, which will pull these back. So this is all we're doing. If you look at the difference from that to that, placing that null line correctly on the diagonal. But the biological upshot is that if we look at the differentially expressed genes, we move from this to this. So here, from the standard way of applying this, we move from nearly 3,500 upregulated genes before to only 210. Those 210 have a really interesting biological connection that we can pull out, and that gets clouded when we mix in all of these mispredictions. So the exploration here allows us to find something that was really sort of obscured before, not because of a mistake in the software, but just because we hadn't explored well enough.
Simon, that's incredibly interesting. I think we do have to move on to Q&A.
Okay.
But it was really, wonderful to hear about all
we
these.
have one final example, but
yes, this will be for the next time. So so the question we've got from the chat from Vicky, I'll just bring it up here, was just really kind of a biological perspective. As a biologist, would she need to work with a bioinformatician to use those methods that you've developed, or would she actually be able to work with it herself?
So the methods that we've got here are no harder to implement than the processing that you're going to need to do. So they're essentially the same degree of complexity to run as the software that you're going to have to run to do the trimming, mapping, whatever. So if you can do that, you can absolutely do these. And the computation for them is decidedly less. So yeah, absolutely.
I see. No, that's really interesting, and it kind of links to the question which I wanted to ask, which is really just around, would it be possible to go even a step further and actually automate those checks so that editors of journals could run them at the time of submission, and actually it could become effectively a part of peer review. Do you think that would be possible or desirable?
Yeah, so some are. So some require quite specific knowledge about the samples. So if you need to know, but certainly something we've looked at is just automating the collection of this data. So not necessarily that you could flag that it's right or wrong. So, but in the same way as MultiQC does, that you can see that set of results all in one place, so that a quick glance down it would allow you to sanity check that very quickly.
All right, wonderful. Simon, thank you so much for giving this talk. I think this field is definitely underappreciated and incredibly important, because without knowing that our data is good, what can we really tell about the biology that we're studying?